semester has been so crazy already.
Tell me about it. I'm just so thrilled that we're juniors. I just feel so much older and wiser.
Same here, man.
What's your favorite course so far?
My favorite course has got to be Writer's BioChem course.
Oh my gosh, me too. It really gets you thinking.
Seriously, junior year is the year for philosophical thought. Like, what is a model protocell, and how is it even driven by encapsulated catalysts?
Funny that you asked that. I have just the article for you. Now introducing Sozak's research paper.
Competition between model protocells driven by an encapsulated catalyst.
Hi there, I hope you're all having a good day. Today we will be discussing competition between model protocells driven by an encapsulated catalyst by Katarzyna Edamala and Jack W. Sozak. The supporting information for this presentation comes from Bioenergetics and Life's Origins by David Deemer and Arthur L. Weber. And the presenters you'll be hearing from today are Magnessa, Catherine, Elena, Fatima, and Liv. So to get us started, I'd like to just introduce kind of the overall question, which is how could protocells that lacked complex systems seen in modern cells evolve to include enhanced vesicle growth? So protocells, as we know, were simple cells containing genetic material enclosed by a membrane consisting of a lipid bilayer, which were often left to compete with one another for limiting resources. And from an evolutionary standpoint, these are often looked at as kind of the origin to how we have the complex cells we do today, which introduces the question of how we made the jump from these simple cells to the complex cells we see today. So one of the ways that this paper discusses that topic is through vesicle growth within protocells membranes. Now, the benefits of vesicles within these protocells membranes is that the cell has control over what is going to be coming in and out of it before we see the sophisticated channels and pumps embedded into membranes we do now. In this paper, we see that this is done through a catalytic process, including the catalytic synthesis of acetylphenylalanine leucinamide, which improves the localization of the membrane and allowed for vesicle growth within cells that were in solution with the catalyst, as opposed to cells that did not see the same growth that were not in this catalytic solution. So in Sozak's approach, we see that their model protocells developed in the lab would undergo various manipulations, such as the addition of the catalyst acetylphenylalanine leucinamide, which would then allow for the observation of how these protocells compositions may have changed to allow for enhanced performance. Now, figure 1 shows us kind of a picture of how this may have happened. And we see the catalytic synthesis of acetylphenylalanine leucinamide, which in solution with these protocells would embed itself into the membrane, which is depicted by these red membranes, and the cells that do not have these, or the catalyst embedded to it, are the gray ones. So the cells with the red, as seen in B, were able to go through processes and grow, whereas the cells without this catalyst shrunk. Then in C, we see how when my cells are introduced, the cells again with the catalyst embedded into the membrane had vesicles that allowed for growth and replication, and the cells without did not. So furthermore, in this presentation, you'll be hearing about the methodology, results, discussion, and critiques involved in this paper. So yeah.
Methods of driving vesicle growth. The first method used is by using the known catalyst surhis in the synthesis of acetylphenyllucine NH2 in order to characterize the changes that compound makes to the growth of a vesicle. This is first done by growing vesicles containing Lu NH2 and acetylphenylethoxy, which are the reactants to form acetylphenyllucine NH2. Then Sir-His is added to those vesicles, increasing the concentration of oleate vesicles in each trial in order to test the amount of acetylphenoleucine NH2 made. This is then compared to the same experiment but done with the other catalyst Sir-His Gly instead. Sir-His is also added to vesicles containing acetylphenoleucine NH2 in order to test the hydrolysis of acetylphenylethoxy, similarly increasing the concentration of OA in each trial. Vesicle membrane interaction and growth of acetylphenoleucine NH2. In order to measure the interaction of acetylphenoleucine NH2 with the membrane, the fluorescence of a tagged membrane probe is used. The concentration of acetylphenoleucine NH2 was increased throughout trials in order to measure the effect on the fluidity of oleate membranes and the effect on fatty acid dissociation. FRET, fluorescence resonance energy transfer, is used to measure the growth or shrinkage in surface area of cells. Here, vesicles that either do or do not contain acetylphenoleucine NH2 are incubated together, and FRET measures the competitive growth between the two. This test is repeated when micelles were added, which act as a food source to the vesicles. To test the competitive growth of the two vesicles, they are put in buffer solutions with varying concentrations of sodium chloride and TMAC, but over a 0.5 molarity NaCl, there seems to be little change in the growth of a vesicle. Does acetylphenoleucine NH2 cause the growth of filamentous vesicles? This is tested by mixing vesicles with 10% molarity acetylphenoleucine NH2 with 100 equivalents of oleate vesicles, which is then gently agitated with a surface area of cells found using FRET. Arginine is used to test the change in a pH gradient across a membrane. Arginine lasts much longer, about 16 hours in the cell, than H+ or Na+, which is typically what causes the gradient. So two solutions of arginine buffer are used to monitor pH of a membrane, one that has acetylphenoleucine NH2 and one that does not. Comparison of catalysts. In order to compare the catalyst Lou-His or Lou-His Gly, vesicles were created with each catalyst respectively. Then they were incubated separately after coming into contact with a substrate with Lou NH2 and acetylphenylethoxy, and then they were mixed and incubated again. Finally, the surface area was measured using FRET. This was similarly done with the catalyst vesicles being given large amounts of feeder oleate vesicles, being kept completely separate, then having their surface area measured using FRET.
So why does this paper matter? One way of explaining how evolution occurred is through model protocells, and Sosak's paper highlights why the model protocell is advantageous in a plausible means of evolution. So the first main result is that the Sir-His catalyst produced more acetylphenoleucine NH2 in the presence of these oleate vesicles. This increased substrate to product conversion from 25% to 44%, which had a diminished substrate hydrolysis of 65% to 10%. So you might be asking why Sir-His is a catalyst. The reason why is because Sir-His glycine is less effective and it only produces half as much dipeptide, specifically 27% versus 44%. The second main result is that competition results in adaptations that are beneficial to protocells. Sir-His remains localized within the vesicle, and the dipeptide exchange only occurs slowly between these vesicles when prepared from oleic acid. So here's our first figure. For part A, the x-axis is time in hours, and the y-axis is the percent yield of acetylphenoleucine NH2. And as the OA concentration increases, there's a faster synthesis and a higher percent yield. And as you can see, at 50 moles of OA, this produced the highest percent yield, and no catalyst produces the worst. So in part B, the x-axis is time in hours, and the y-axis is the percent yield of acetylphenyl OH. And as you can see, the AC-Phe substrate is being hydrolyzed less in the presence of these oleate vesicles, and there's no OA at the top, which is the most hydrolyzed, versus 50 millimoles of OA at the bottom, which is the least hydrolyzed. In graph C over here, the x-axis is the oleic acid vesicles in millimoles, and the y-axis is the percent yield of acetylphenoleucine NH2. What this is showing is the yield of the dipeptide versus the concentration of the oleic acid vesicles. And then note that there's an increase in millimoles of vesicles as you have a larger percent yield. And then our last one, which is D, the x-axis is the oleic acid vesicles in millimoles, the y-axis is the percent yield of acetylphenyl OH. And what this is showing is the yield of the hydrolyzed substrate versus the concentration of oleic vesicles. And note that as the millimoles of the vesicles increase, the percentage of the substrate being hydrolyzed decreases. So the third main result is that the dipeptide might drive vesicle growth at the expense of pure fatty acid vesicles. There's a decreased fluidity of oleic membranes, and there's a decreased fatty acid off rate and a decreased desorption rate. The fourth main result is that when incubated together, vesicles containing acetylphenoleucine NH2 grew at the expense of those without, and the surface ionic interactions actually affect these fatty acid exchange. So NaCl in self-buffered cells will actually reduce or prevent growth, and this is because the more lipophilic peptides induce greater competition. So if you look in this next figure, you can see right here in figure 3, the x-axis is the dipeptide of acetylphenoleucine NH2 in the percent mole, and the y-axis is the final over initial surface area. In A, you can see that with acetylphenoleucine NH2, there's showed growth, and this is in the shaded areas. And without the dipeptide, there's actually shrinkage, which is the clear areas. In B over here, you can actually see the larger surface area and more vessel growth, with the peptide, which is shaded, as opposed to those without, which is the clear region. And over here in figure 5, the x-axis indicates if the buffer is present and what salt is present. The y-axis over here is the final over the initial surface area. Note that the buffer leads to a larger final over initial surface area ratio, and salt prevents growth, which is the clear column shown over here. Fifth main result is a proposed mechanism of division. So it results in these fragile thread-like structures. They break off and form daughter cells. And then the sixth main result is that the dipeptide is synthesized more in vesicles with Serhis, and you get a 28% yield. And the vesicles containing Serhis have an increase in surface area by 24%. So right here, you can see the thread-like structures and then new daughter cells being formed over here. So the seventh main result is that vesicles that contained acetylphenoleucine NH2 developed a transmembrane pH twice as large as the vesicles without a dipeptide. And the eighth main result is that vesicles containing Serhis have an increase in surface area. When livestock is available, both grow, but Serhis grows 2 times more. So as you can see here, without the acetylphenoleucine NH2, there's a much higher pH, which is a more basic solution than as opposed to down here.
This research paper is looking at Darwinian evolution and different heritable variations which are required for fitness. So in this experiment, the protocells were chosen to look at what processes would lead to evolution in protocells. And the reason why protocells were chosen are because they have relatively simple physical and chemical processes. And they can also, their growth is dependent on fatty acid membranes, which are easier to measure in terms of size. There was a catalyst, encapsulated catalyst, which was used to drive vesicle growth. And that was done through the catalyst synthesizing a dipeptide called acetylphenylalanine leucinamide. I'll just call it dipeptide for the ease of pronunciation throughout. The dipeptide further caused a competitive vesicle growth. And that can be compared to like a regular predator-prey relationship, competitiveness for nutrients, for example. Also in the experiment, there were 2 types of catalysts which were chosen. There was the serine histidine complex and then the serine histidine glycine. But it was observed and mentioned in the results section that there was a greater dipeptide synthesis with the serine histidine complex as compared to the serine histidine glycine complex. One of the reasons also why the dipeptide is adaptive is because it allows, once it's causing vesicle growth, it stays inside the cell, and the way it causes vesicle growth is that it attracts components inside the cells, and then they build up and cause growth. The growth was also compared between the 2 serine histidine and serine histidine glycine through measuring the pH.
Now moving on to the critique, including major points from the paper. The first one, importance. Why is it important to study protocells and how did the paper show it? The first reason the paper is important is because it discusses Darwinian evolution. The article talks about different protocell models and how it competes for growth. The protocell will adapt for survival depending on the environmental conditions, such as fatty acid vesicles and the presence of specific catalysts. It leads to natural selection based on enhanced fitness for survival. This process of protocells competing for growth ultimately leads to modern cellular life and modern biology. The second reason the paper is important is because it suggests an origin of protein enzymes and peptide formation. Since amino acids are the building blocks of proteins, the formation of the dipeptide cerylhistidine is occurring somewhere in the protocell. In favorable conditions, such as high presence of fatty acid vesicles, cerylhistidine catalyzes a higher yield of peptide formation and synthesizes N-acetyl-L-phenylalanine glucosamine, which drives competitive vesicle growth. The peptides may have been synthesized by ribozymes. This leads to the modern RNA compartment in the cell being the ribosomal unit where protein synthesis occurs involving RNA and amino acids. The second point is improvements. Protocells suggest an RNA world hypothesis that we will talk about, followed by additional experiments that could provide supplementary information. The RNA world hypothesis is the proposition that RNA was originally the genetic material, since it is capable of self-replication and can act as an enzyme, like ribozyme, to catalyze chemical reactions in primitive cells. The article could be improved by maybe involving some background of what the RNA world hypothesis is and what it includes. This would give a background of the formation of peptides mentioned in the text. It does do a good job of suggesting the potential of RNA and its major contributions by linking it in the abstract and discussion parts of the paper. An additional experiment that could be performed is a northern blotting analysis. This could be done to compare the amount of RNA between the cerylhistidine catalyst and the cerylhistidine glycine catalyst. The purpose of a northern blot is to study the size and steady state level of RNA in a cell. If high levels of RNA are detected in the cerylhistidine catalyst protocell, then it suggests or confirms enhanced vesicle growth and increased surface area. The third major point are issues or some matters regarding the experiment, which include types of catalysts used, which are dipeptide cerylhistidine and the tripeptide cerylhistidine glycine. The control would be no addition of catalyst and seeing vesicle growth by itself. Some limiting factors could be the amount or type of feedstock provided and selective methods used. Overall, the data provided in the figures represents the results in an organized manner. One thing that could be added would be to include the tripeptide in the graphs that show increased surface area for visualization purposes. Please come to class with one or 2 points you took away from the paper and ready to discuss. Thank you.
Wow, I just loved this research paper.
Life sure is interesting, right?
If only someone could present this material to our BioChem class, I bet that they would get 100% and tons of brownie points.
Oh, for sure.